Happy morning, dear children of class 7, and I am glad to welcome you all to one more botany class. What are we going to learn today? Our today's topic is going to be about chloroplast. So what is today's topic? Chloroplast. In our previous few classes, we have been studying about plants, right? Since the name of the lesson is plant life, we are studying about plants, and in our last class we have studied about the process of photosynthesis, and in today's class we are going to learn about chloroplast, which is an important component of photosynthesis. So let's get started. Before learning about the detailed structure of chloroplast, let's know what is a chloroplast. What is a chloroplast? A chloroplast is a organelle. What does this mean? Organelle. Have you heard this word organelle before? What is an organelle? See, this is a structure of a plant cell, and either a plant cell or an animal cell, all of them have small structures in them called organelles, except for the nucleus. Every cell has nucleus, you know, right? So where is the nucleus here? So this is the nucleus, and you have a fluid filled in any cell that is called as cytoplasm. So except for the nucleus and the cytoplasm in a cell, all other structures, you have many structures here. Now see, you have some red color structure, some black color structure, some yellow color structure, some green color structure. All of them inside a cell are called as organelles. The small structures inside a cell are called as organelles. So this chloroplast is one such organelle, which means there are many organelles, and chloroplast is one of the organelles present only in plant cell. See, the other organelle, say for example mitochondria is an organelle, endoplasmic reticulum is an organelle. You will be learning about all these in the upcoming classes. In the same lesson we have the explanation part, we will be learning in the next few classes. Clear? So there are many organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, all, which is common in both plant cells and animal cells, which means some organelles are present both in plant cells and also in animal cells, but this organelle called as chloroplast is present only in the plant cells. Clear? So this is a plant cell, and where is the chloroplast here? Can you see some oval structures, some green color oval structures, not the red one, only green ones? These are called the chloroplasts. So because of these green color chloroplasts only, the plants are green in color, because every cell has this green structure called as chloroplast, and only this chloroplast, which is green in color, gives the green color to the plant. Since every cell has some green structures, overall the plant is also green in color. It is absent in animals, so animals are not green in color. So what is a chloroplast? Chloroplast is a organelle in a plant cell. What is the shape of a chloroplast? The shape of the chloroplast, already I told you, oval. What is oval? Like almost the shape of a egg, right? Oval in shape, not a circle, but it is oval in shape. Where can you find chloroplast? The part of the plant which is most green in color is the leaves, right? When you compare to the stem and other parts, all, which part is most green in color? The leaves. So the leaves are green in color because they have more chloroplasts when compared to the other parts. But other parts like young stem, what does this mean? Young stem means when the plant is young, the stem which is formed are green in color. Now see, out of a seed, when a plant germinates, the stem is green in color first, but as it grows it becomes brown in color. No? So during the stage when it is green in color, it has more chloroplasts, that's why it is green in color. And also in some microorganisms like algae. Algae is a lower kind of a plant, a primitive plant you can say. So this algae you have studied, no? Spirogyra, Chlamydomonas are all algae. They are also green in color because of the presence of this organelle called as chloroplast. Clear? So Where can you find? In leaves, in the young stems and in the algae. So what have we studied about chloroplast? Chloroplasts are organelles and they are oval shaped structure, and you can find them in the leaves, young stem, and also apart from plants you can find in other lower plants such as algae. Clear? Now we learn about the detailed structure of a chloroplast. What are all exactly present in a chloroplast? See, in the previous slide I have shown you structure like this, no? Like oval structures are chloroplasts. We have learned what is present inside the oval structure. All these parts are present inside the oval structure. We learn about that in detail. So initially there are two membranes called the outer membrane and the inner membrane. Which is the outer membrane here? This is the outer membrane. This is the outer membrane and this is the inner membrane. So the chloroplast has one outer membrane and one inner membrane. In between the outer membrane and the inner membrane there are some spaces. There is a gap between the outer and the inner membrane, and that space or gap is called as intermembrane space. Always the word inter means between. The word inter means it is between. Clear? So inter means between. So between outer and inner membrane, the space is called intermembrane space. So now you know there is outer membrane, inner membrane, and between these two you have intermembrane space. Then what all you can find? Inside you have a liquid like structure, just like how cytoplasm is present in every cell. Inside the chloroplast you have a liquid like substance which is called as matrix. This substance is called as matrix or stroma. So stroma is also called as matrix. So inside the inner membrane you have a liquid portion which is called the matrix or the stroma. Matrix or stroma. And then you have some coin like structures. Can you see here? Throughout the stroma you have round structures which are like a coins, and these coins are called as thylakoid. All these terms are new to you. Spell it again and again for 3-5 times, then you can get those names in your mind. Clear? So what are the coin like structures called? Thylakoids. There are many thylakoids packed on one another, and thylakoids together are called as granum. What does this mean? See, if this is one thylakoid, there are many thylakoids arranged one above the other, and this is called as granum. Clear? N-U-M. So one coin-like structure is called thylakoid, and a pile of thylakoids stacked above each other, it is called by the word granum. So what have we studied? Outer membrane, inner membrane, intermembrane space, a liquid-like structure called stroma or matrix, and inside you have coin-like structures which is called thylakoid, and the thylakoids packed on one another, that structure is called as granum. And two of such grana, singular is called granum and plural is called grana. Okay, suppose if there is a one granum here and another granum here, these two grana are linked together like a bridge like structure. Okay, and that bridge like structure is called Stroma Lamellae. What did I tell you? If there are two grana, there is a bridge connecting both the grana, and that structure is called as Stroma Lamellae. What is it called? Stroma Lamellae. So what all you have studied in the structure of chloroplast? Outer membrane, inner membrane, intermembrane space, Then coin-like structure is called thylakoid, and many coins together are called granum, and two grana are connected by stroma lamellae. So all these terms you have to memorize. Clear? So this is all about the structure of chloroplast. So some explanation part. Already I told you there are two membranes called outer membrane and inner membrane, no? So that's why chloroplast is described as a structure which is double layered membrane, a structure with double layered membrane. What is double? Double means two, no? So chloroplast is a structure with double membrane, and the membrane surrounds the stroma. What is stroma? I told you, just like the cytoplasm in a cell, the liquid portion in the chloroplast is called stroma, no? So where is the stroma? So the entire portion is filled with stroma. So this is all filled with stroma. So the membrane surrounds the stroma, which means this membrane covers or surrounds. It is the covering of the stroma. That is what is meant. Then the stroma or the matrix is the ground substance. What is ground? Ground means it's not grinding. This ground refers to as the base of the chloroplast. It is the basic structure of the chloroplast, which means there is stroma, and inside the stroma only you find all the other components, such as what all you have inside: thylakoid, lamellae, and grana. All you have, no? Everything is present in this stroma only. That is why it is called as the ground substance, which means it is the basic substance in a chloroplast. Clear? And then in the matrix you can find thylakoids. In the stroma only you find thylakoid also. Already while explaining the structure I have told these points. Anyway, in the explanation part of your book these facts are given, so I repeat again. So in the structure of the chloroplast, you know that it is double layered membrane called outer and inner membrane, and the membrane surrounds the stroma. There is stroma in the middle portion, and the membrane covers the stroma, and the stroma or the matrix is the ground or the base substance, and the matrix has thylakoids, which is coin like structures. Clear? So this slide is explanation part about thylakoids. This also I have explained while explaining the structure slide. Here again let's learn some more points. What is thylakoid? I told you the coin like structures present in the stroma is the thylakoid. Which is the thylakoid? So these are thylakoids, right? So these are thylakoids. They are flat. How it is? It is not like concave or convex. The surface is flat and it is sac like. What is sac? Usually at home we use sac, no? What is sac? What will be inside the sac? The sac is usually empty. Inside this coin like structure it is empty. That's why it is called by the word sac. So what have you studied about the structure of thylakoid? First of all, it is flat. Then it is a sac because there is nothing inside, and then the thylakoids are interconnected. Interconnected means every thylakoid is not a separate structure, all are linked together. So these thylakoids are all linked together, as well as thylakoids together. I told you granum, no? It has a term called as granum. The grana are also linked together. So thylakoids are interconnected. One thylakoid and the next thylakoid are connected for the transfer of materials all. So they are not separate elements, they are interconnected, and they are connected by stroma lamellae. Which is stroma lamellae? Do you remember? So this is the Stroma Lamellae. What is Stroma Lamellae? This is one granum, this is one granum, and two grana are linked by a bridge, and the bridge is called as Stroma Lamellae. Clear? This is about the structure of chloroplast. Then what is chlorophyll? You must have heard about this word chlorophyll, right? So what is the difference between chloroplast and chlorophyll? Chloroplast is organelle. I told you already. Chloroplast is a organelle. Then what is a chlorophyll? Chlorophyll is a pigment which gives color. What is a pigment? We have melanin pigments, right? What is melanin pigment? The black pigment which is present in human body is called melanin. Similarly, the green pigment which is present in plants is called chlorophyll. So chloroplast is an organelle, and chlorophyll is the pigment which is the one which is responsible for giving coloration. So chlorophyll is a pigment, and where it is found? It is present inside the thylakoid of the chloroplast. What is thylakoid? The coin-like structure is thylakoid, right? So this pigment, that is the chlorophyll, is present in the thylakoid of the chloroplast. That's why the chloroplast is green in color, because of the presence of chlorophyll pigment. Because chlorophyll pigment is green in color, and since it is present inside the chloroplast organelle, it is also green in color. So what do? They absorb wavelength which is red and blue in color. This pigment, they capture the red and the blue color, and they give out green color. They reflect the green color. So whatever color is reflected, that color only can be seen with the eyes. The color what they absorb, we cannot see what color they absorb. This pigment has a special property to absorb the red and the blue color, and it will reflect the green color. That's why we can see green color. The chlorophyll appears green in color, as well as the chloroplast organelle also appears green in color. This is the reason why chloroplast and chlorophyll are green in color, because they reflect the green color. Clear? And then this pigment, chlorophyll, has a special property to trap the light energy. Chlorophyll is essential in photosynthesis. Chloroplast or chlorophyll pigment is essential in photosynthesis. Why? Because the light energy is trapped, which is attracted or pulled to the plant only by this pigment called as chlorophyll. They trap the light energy and initiate the chemical process. Which chemical process? The process of photosynthesis. So for photosynthesis to happen, you need light energy as one of the component, and this light energy is trapped by the chlorophyll pigment. And then after photosynthesis, what is produced? Food is produced. What is food here? In the plant, the food is the glucose, and this glucose is stored in some places like the roots of the plants, the stem of the plants, or in the leaves of the plants. Clear? So what have we studied? Chlorophyll pigment has a special property to trap the light energy, and it will initiate the chemical process of photosynthesis. So after photosynthesis, the product formed, that is the food or glucose formed, is stored in three places. Which are those three places? Roots, stem, and leaves. Clear? Then one activity is given. Actually we can do this in lab, but since you are not here, I'll explain like a theory. Once you are back to school, let's go to the lab and do this. Clear? So for this activity, why we have included this activity in this topic of chloroplast? This activity, the aim of this activity is to prove that chlorophyll is essential for photosynthesis. So what are we going to prove through this activity? Why you do an activity? To experiment something or to give a proof of some fact only we do activities, right? So from this activity we can understand that, or we can prove that chlorophyll is essential for photosynthesis. Clear? So for this activity what you are going to do, you are going to take a variegated leaf. What is a variegated leaf? See this leaf, this is a variegated leaf. What can you observe in this leaf? Some parts of the leaves are green in color and some are yellow in color. No? Some part is green in color, some is, there is variation in colors. That's why it is called variegated leaf. But some portion should be green. In many crotons, no, what you grow at home, you can see this variegated pattern. You will have like red and green combo, or white and red, green and red, like that all you have combinations, green and yellow, like that all you have many patterns in croton plants what we grow for decoration purposes, right? In front of the house all we used to place, no, those are all variegated leaves. So here we have chosen a variegated leaf which is green and yellow in color, and you are going to de-starch it. De-starch means removal of starch. Already in the plant there will be starch because of photosynthesis. The leaves will be having starch in them. You are going to de-starch. De-starch means remove the starch. How can you remove the starch? Can you cut the leaf and remove? No. Just keep it in a dark room for 48 hours. That is for 2 days. Just keep this should be a potted plant, because if it is a garden plant you cannot keep it in a dark room, no, for 48 hours. That's why it is given that choose a potted plant so that you can carry the pot to the place where you want. So this variegated leaf should be in a pot, it should be in a pot. So take the pot and put it inside a dark room for 2 days. Okay. So after you put, what will happen? The starch will be removed, because our purpose is de-starching. No, there is no starch. There is one fact. How will you know there is starch or not? How can you test? There is a simple experiment. See, whenever you add iodine solution, what is that? Iodine solution. So starch plus iodine solution. This solution is usually brown in color. Iodine solution. So starch and iodine solution will give blue, dark blue color. Dark blue means it is actually called as blue black. Blackish blue. The blue is so dark that it appears almost like black. Okay. So what did I tell you? Whenever there is starch plus iodine solution, you will get this blackish blue color or blue black color. So if I take a leaf and pour iodine solution, if there is starch in the leaf, it will be blue black in color. If there is no starch, it will be brown or some other color, but not blue in color. So what did I tell you? I told you to de-starch a plant for 48 hours. No. Now if you take a leaf and pour starch on the plant, on the leaf, what color you will get when there is de-starching? You will not get the blue color, because the starch has been removed after keeping it in a dark room for 48 hours. Okay. So now I have de-starched the plant, no? De-starch the plant, so there is no starch. Suppose if I pluck a leaf from this de-starched plant and pour iodine solution, will it be blue black in color? No, it will not be blue black in color, because there is no starch in the leaf, right? So initially after de-starching, if I pour iodine solution, there will be no blue black color, because there is no starch. Now what you are going to do is take the potted plant and keep it in sunlight for 5 to 6 hours. Okay. So first you have de-starch. Now take the same plant and keep it in sunlight for 5 to 6 hours. So now what will happen? The plants begin to undergo the process of photosynthesis again. So whenever there is photosynthesis, again starch is formed. No, starch is the product of photosynthesis, which is the glucose. So now starch is formed, and now if you take one leaf and if you add iodine solution, you can find blue black in the center region, which was initially, this is the initial leaf before destarching and before keeping up it in 5 hours of sunlight. Okay. This is the initial leaf which was kept in the dark room for destarching. Initially it was in this color, but now after destarching and after keeping it again in sunlight for 5 hours, you can find the blue black color in the center, and these portions will not be blue black in color. Why? Because this portion was already yellow in color, which means here there was no chlorophyll, but this is green in color because it has chlorophyll. So I told you, whenever there is starch and iodine combination, you will get the blue black color. Here this portion is blue black color, which means this portion has starch, which indirectly means that only the green color portion has the ability to undergo the process of photosynthesis and make starch. So whichever portion of the leaf is green in color will make starch by the process of photosynthesis, and these portions cannot make starch or they cannot undergo photosynthesis. That's why it is not blue black in color, it is red in color or brown in color of the iodine solution. So this experiment proves us that photosynthesis will happen and starch is produced only in the places where there is chlorophyll. So chlorophyll is the one present in the green portion, and that's why from the result we have got the blue black color. This is a proof that chlorophyll is present and it is responsible for photosynthesis in the green portions of the plant. And for your homework, you are going to read pages 71 and 72. Clear? So read your daily lessons so that you are thorough with the topics what I teach. So that is all for today's botany class. Until I meet you in your next zoology class, you all take care and bye bye.